So, thank you very much. Hope everybody had a good lunch. Now we're going to talk about something else that also may be much more relevant to psychiatry. We're going to talk about clinical biomarkers, clinical measures used as biomarkers. So just to remind you of some of the questions that you might want to be thinking about during the presentations, we're interested in learning what are the best cognitive measures to detect change, or best measures. It just so happens that most of the presentations are about cognitive measures, but be thinking about clinical measures in general. What are the most robust and reliable measures for selecting patients to enrich clinical trials? Now, we know that different psychiatric disorders have different cognitive impairment patterns. So is it appropriate to use similar or the same cognitive measures across diagnoses? How rigorously should we look at test, retest reliability, and utility of markers? As Jamie alluded to this morning, we actually often use markers that were conceived of as diagnostic markers as endpoints in measuring change. And is that appropriate? Do we need to consider that when we look at clinical markers for endpoints and as biomarkers? So I'd like to ask Bill Potter to come up and introduce our speakers.
I'll do it from down here. And our first speaker is Stephane Pollentier from Boehringer Ingelheim. And as he's coming up, Stephane has been a leader among industry scientists over the years in trying to advocate for broadening our suite of what I'll call clinical measures, but measures that might tie into a domain which might have some underlying biology and circuit function. So we contextualize this in terms of cognitive measures, but I'm hoping Stephane takes us even beyond just that. So Stephane, please.
Yeah, actually I'm completely going to avoid cognition, to be honest. But at the moment, I'm also avoiding slides, and so I'm sort of wondering whether technically we can get this set up. I don't know whether people online can see the slides. I'll just give it a second. There we go. Great. So I was given the title Task-Based Behavioral Markers, and from that perspective, as a full-time employee of Boehringer Ingelheim, which is my disclosure, I sort of figured I would address this in the following way. I speak a little bit about tasks and paradigms and performance measures and drug development trials, in particular in psychiatry, because that's what I do. I'm head of CNS medicine for Boehringer, and we oversee clinical development in psychiatry. We're not in neurology. I'll give you a few in-house examples of the sort of tasks and paradigms that we've built into our own ongoing studies. And I definitely want to acknowledge that many companies do the same. But I sort of lacked the time to call my colleagues and ask for slides that I could use in the context of this session. And the reality is that much of the stuff that is ongoing now is not yet published. It's typically posted or people speak about it in congresses, but it's not published yet, so it's a bit difficult to get your hands on their data. I think a big question is all of this stuff ready for prime time? That's a question that keeps coming back, so I certainly want to address this and some of the gaps and ways to address them. And this has been a great 1.5 days, and I also enjoyed coming back because the reality is that a couple of years ago, as Rosa may well remember, and some others too, we tried to launch a project through the Biomarker Consortium, and at that point in time didn't find traction. But and without bitterness, I'm happy to say that we've succeeded in getting it off in Europe. And I'll speak to how we set it up and what we're doing there. So it was mentioned before that traditional endpoints in psychiatric drug development trials are qualitative measures. And that's good, I think, because you want to talk to patients and hear how they feel. That's how are you doing? That's basically a very qualitative question. And we've structured it and have registered drugs based on those qualitative endpoints, so they have their value. And if you talk about patient-reported outcomes, which typically is a big issue in many of the more somatic indications, I always felt that in psychiatry we do nothing but patient-reported outcomes in our pivotal studies. But they have problems, and they're all well known to all of you: subjectivity, bias, variability, etc. That's the window of opportunity, I think, to corroborate these qualitative measures with quantifiable measures of performance. And these could be focused tasks that actually engage an interrogated circuit that has been more or less established in some of the fMRI settings or some of the other more biological-driven assessments. But there are also higher order tasks, and I believe they have a considerable value because they really approach what is called functional capacity. One of the main things I think we struggle with in drug development in psychiatry is that people sort of understand with relatively modest, if not even weak effects we have with our no drugs, and it's not reasonable to expect that we would have a breakthrough drug for one of the indications. It's always going to be subtle effects. And so the question is, what is the clinical relevance of that? What is the meaning of improving a couple of points on a cognition scale? And Rachel will speak to cognition later on. So the way I look at it is that behavioral tasks or performance measures serve a great purpose, in particular in early stage translational medicine settings. And I'm sure you're aware of the fact that most companies now will not engage in large scale trials and investments without establishing target engagement on one hand, but also some form of pharmacodynamic evidence. Fully understanding that there is no predictive biomarker or test that will help us guarantee that the effect that we've seen translate in a clinical meaningful effect down the road. But I think in contrast to the past, where we believed the theories of our pharmacologists and sort of jumped into large-scale studies because it sounded so convincing, that these days are gone. And so behavioral tasks that have typically been developed, a lot of them in the context of fMRI or parallel to other measures, are very much used in that early setting. But I'm a late-stage developer, although at the moment we don't have much late-stage development ongoing. But I'm sort of looking at this from the more, well, if I see something in a task in an early setting, can I take this forward to help me guide my further development? Can I yield supportive evidence out of these tasks? Can I identify subgroups? And even in the extreme end, could these become measures of functional capacity? In other words, I was really considering whether I would call this talk, the quest for the 6-minute walk test in psychiatry. And I was very pleased to see that in the opening session, in the presentation of the FDA, the 6-minute walk test was actually right on there on that board. It is low tech, it's simple, it has face validity if you wish. And it's measures response to medical interventions. It's used as a one-time measure of functional status, and it's correlated with morbidity and mortality. So what more could you want of a surrogate task? So the question is which tasks and when to use which task where. And I don't think that the intent of this talk was to give you an overview of what would be the best task. I think the ad hoc approach, and I really appreciate the National Advisory Mental Health Council work group and the report they created, really did an excellent job of reviewing all the tasks for all the different domains and subdomains in RDoc and all the evidence that was available at that point in time. And since the report was published in 2016, it means they reviewed data from way before there. And so if you sort of, well, what is your wish list? Certainly would wish that in 1 or 2 years this is repeated and updated with the data we've generated in the meantime. I think that would really be extremely help, because one of the things we do in industry is look through the literature. We go to consultants, yes, and they help us, but consultants sometimes are also a little bit biased with respect to the work they do themselves, and you go to one and you go to the other and you sort of get conflicting input, and then you have to decide. And then it's nice to have this sort of independent assessment and that can help you make choices. And in the context of our previous project, we actually also did a very nice workshop that also helped pin down a number of tasks, in particular in the context of reward processing and motivation. So what I'll do now is give you a couple of examples of what we're doing in our studies at the moment, in phase 2 studies that are ongoing, large phase 2 studies. And again, with the disclaimer that I know that a number of my colleagues do similar things in their programs. Okay, so this is a quite large phase 2 study that is set to demonstrate clinical proof of principle, if you wish, and to also help identify an effective dose. And I'm going to show you a little bit in more detail some extracts of the poster that we recently had at CERS. Our approach has been that for phase 2 studies and pivotal type of studies, we will go with the established regulatory endpoints. That's our main objective, is to make sure that we can deliver a drug in a way that is expected based on the data that are required. But we feel that we can explore, alongside the main objective of our clinical studies, new tasks. And one of the things, so the study is in cognitive impairment in schizophrenia. Sorry, I should have made that clear. But one of the things we feel is important in that particular setting is to see to what degree motivation plays a role in the performance on some of the cognitive tasks. And so one of the tasks that we've included is the balloon effort task. This is Jim Gall's task developed where you have a low potential reward and a high potential reward, and then you make a choice whether you invest more or less effort. A task that I'm not sure has been used in fMRI, but it certainly has been used in schizophrenic sessions, patients. And then the other one we've added as an exploratory task for the purpose of this study is the probabilistic reversal learning task, which addresses reinforcement learning as an important component of motivation and learning. And this is a sort of shift task where once you figured out which one of the two symbols is the one that is rewarded and have confirmed this by giving correct answers for a while, the rewarding is shifted and you have to figure out if you can do that. And it's established in the literature that schizophrenic patients have issues in performance with this. And that's basically what we confirmed in the baseline snapshot of this ongoing study. And by the way, as I think I mentioned, we do this typically in sub-studies. In this particular case, we only do it in the US. That has simple logistical and pragmatic reasons, because if you do it on an international scale, and this study runs internationally, you're immediately confronted with translation, with things such as dollar signs for the reward of some of the tasks, which are not accepted in other countries because we pay in euro. So what the heck is the dollar sign worth? And then there are even issues, because for some of these tasks, participants actually get paid out. And so there are ethical committees that then raise the concern, well, if someone suffers from not being able to conduct well in this task, you're actually punishing him if the reward would be equivalent to the performance in the task. So you sort of have to figure out a way of rewarding people for a different performance, even if it's actually at the heart of the task that they would be performing better and therefore get more rewarded. So what we've seen in our baseline assessment here is that basically the performance on the task is what one would expect in comparing it with literature. But of course, for us the question is are these tasks drug sensitive? So what will happen in the placebo arm versus the treated arms? Will we be able to see an effect at all? And if so, how does that correlate with the true primary endpoint of the study, which is cognitive assessment? And we repeat this in another study that we just started, where we will be assessing again in cognitive impairment and schizophrenia the effect of our drug on the background of standardized cognitive training. So we repeat the balloon task, and then we also introduced the virtual reality task. This is the VerveCAT developed by Rich Keefe and his colleagues, which is basically a virtual setting where a participant has to cook a menu, detect what is missing in the shelves in the kitchen, take the right bus, go to the shop, buy the ingredients, pay correctly, find the bus back, and then complete the task. And that is a sort of instinctively relevant way of assessing whether people improve in their daily functioning. The team around Keefe, they've done quite a lot of work in terms of standardization of the task, and I fully appreciate that this is beyond what you would call a biomarker. Nevertheless, if you talk about task-based measures, in my view, this is one. So the question is, are these tasks, and there are many, ready for prime time in drug development trials? And I think we discussed issues of construct validity, which is, of course, very challenging in our field because we don't really have good psychopathological understanding of the details. There's the whole battery of psychometric characteristics. I think a very important one, and I made the point before lunch, is the standardization of parameters. Why are there 7 blocks or 5 or 3? Does it really matter? Has anyone assessed that? the duration of tasks. If you want to take tasks into multi-site section, you can't ask people to sit down a half an hour and tap on a computer. The matrix cognition test battery already is more than an hour, and it's really giving us headache. And I know you've developed a very sweet and short cognitive assessment, but unfortunately the matrix battery is the one that finds the highest regulatory acceptance for the moment. As mentioned, in drug development is global, so we're all the time confronted with the cultural and language effect. Pediatric use is mostly lacking in the availability of data in children. In particular, when we look in translational settings, it's extremely helpful to have an analog animal model to be able to bridge. We mentioned normative data also in the context of EEG. And then the big question is, of course, the sensitivity to change and lack of loss of function. We use the tasks that I mentioned before at the moment as standalone, but many of these tasks are frequently used with brain measures. That's what we do too in translational medicine settings, where you have subjects or patients that you can, in a very well-controlled setting, where you can blast a number of assessments on them without which you couldn't do in a large-scale study and a multi-site study. I think an important aspect is copyright and access. I fully appreciate that task developers should receive credit and earn money for the work they invested. But if it gets to a point where it becomes difficult to find that access and to be able to ensure that one can take that task and take it through the remainder of the development, then we have issues. And actually in the project that we set up, we ran into this trap where an exclusive contract was made on certain tasks and then you sort of blocked because that's not the way you want to go. Many tasks are now delivered electronically, which brings a whole bunch of other issues. And from a trial perspective and drug development perspective, most of our data now are collected remotely. And so we have this whole issue of CRF Part 11 compliance, data integrity compliance, that needs to be adhered to. And since many tasks are developed in academic settings and are programmed in R or other open source programming, that's typically, if you then want to start a study and think about, well, I could maybe include the task from this or that academic center, you find yourself confronted with all these things that you need to get settled before you can integrate it in the remainder of your study. So the quality of data aspect is something that at certain points is prohibitive, actually, of including and lobbying internally within the company for inclusion of tasks. I think with that goes all the necessary training that you need to ensure to make sure that the task is done in a multi-site setting in an appropriate way across all sites. And all that speaks to a request in many cases to try to make sure that the task parameters are optimized and the task duration is reduced to an amount that is doable on a large scale. So the question is it ready for prime time? And that, I guess, depends on what you define as prime time. As far as I'm concerned, yes, we include stuff in our studies, and we want to include more and more, and many other companies will do so, and that beyond translational settings. The question of treatment sensitivity obviously is a big one, but well, we'll have to establish treatments, and then we can decide on whether there was treatment sensitivity or not. But what definitely is lacking is tasks in comparison, and I really love this publication from Reddy and Bill Horan and Treadway and many people that are active in this particular field, where they went through the effort of comparing 5 different effort-related tasks in one similar population. So that gives us, in terms of planning and good understanding of the task behavior, we can look at the variability and sort of work with that in the context of our clinical trials. But what you typically see is that tasks are developed by an academic site and are tested in one type of population, not across the CNS spectrum, and certainly not agnostic to diagnostic category. And in most of the cases, it's a comparison, a discriminant validity versus healthy controls. And we sort of lack an understanding how a task behaves across the spectrum. So these are the gaps that we try to address with a project that I'll speak about in a second. But I want to come back to BSNP, which was already showcased a couple of times this morning, and I think it's really a great example of data generation. Although they didn't really have that much tasks in there, but I want to plug the Pursued Eye movement, which I personally feel is an undervalued potential biomarker. I mean, it's historically around for quite a while, actually, and I understand the complexity of assessing it. But I think technology has evolved. There are some gizmos around now, I think, that really help conduct this in a more simple fashion, and I really hope that we can sort of expand on that. In a similar vein, I briefly wanted to mention this European initiative, which, in contrast to BSNP, which you could consider a deep phenotyping around psychosis, this is a deep phenotyping of social withdrawal. It's also a smaller scale study, admittedly. Nevertheless, they have a lot of tasks in there. Again, the reward processing tasks. And I think you figured out by now that I have a weak spot for this type of task. But there's also a number of social cognition tasks. And importantly, I think what is also in the study is a measure of sociability and social exploration by a smartphone application. And I use that also as a plug for digital biomarkers. And I know there is discussion whether you can call digital biomarkers biomarkers. But I can assure you this is a rapidly evolving field. All companies are looking into this because a number of these assessments, these moment-by-moment quantification, this passive monitoring have such tremendous face validity that in a setting of large-scale trials, it doesn't really matter if you have a couple of gaps and data points missing. I appreciate the comment that was made in a phase 1 setting. If you do a wearable, you want to have a very precise measurement. But here, the volume of data, the fact that you can show that people that perhaps were less active in the baseline stage after your treatment walk around much more, have much more smartphone contacts and things like that, I think that that's supportive evidence for improvement. So let me spend a few words on the Reward Task Optimization Consortium, which we've now succeeded in getting started. And the aim was really to qualify a battery of tasks that would measure different reward processing domains and would be qualified for use in multi-site drug development trials. We succeeded in getting funding from 5 partners, but I have to admit it's on a shoestring budget, so it's really not a big investment. And we had support by Pivotal and BioTrial as specialized CROs, one on project management and trial conduct, and BioTrial on EEG measures. And then we have 4 sites in Europe, the Netherlands, Germany, Spain, and Greece, which will also give us the translational and cultural cross-validation, if you wish. We've decided to focus on 3 reward processing tasks and 2 indications or target populations, which, if you wish, and then also use healthy controls, building these tasks to all the necessary regulatory standards and standards of data integrity that we need in our trials, whether it's regulatory required or not. We want to be sure that the data reflect what was observed. We will explore EEG in some of these tasks simultaneously, and hopefully the platform will be usable or reusable for further other tasks. It's a simple study, it's a cohort study where we will pilot the tasks, and then once the performance is in line with what was published in literature, we will move ahead and compare depression, schizophrenia, and healthy controls. And the task constructs, or the constructs we want to cover, are the initial response to reward, effort in reward valuation, and then also a probabilistic and reinforcement learning task. And I can speak to that more if needed. But I have the task details in the appendix, and the slides will be available after the sessions, right? And so with some relief, we can say we are here, which is always a true statement, I guess, in many ways. It took us 4 years to get to this point, just as a matter of reminder here. So it's easy to talk about pre-competitive collaboration. It's a little bit harder to do it, and I realize very well this is not an ATNI type of project, but it goes at the heart of, I think, some of the things one probably need to consider in prioritizing your project's proposals. And part of the time that it took is as usual, contracting, and then certainly if you have 5 pharma partners, it becomes a little bit complex. But it's settled now, and we're ready to go, and we're starting, and we're optimistic we can pull it off in 1 year from now. So closing up with some additional reflections, and I'm going to misuse the context of use sort of connotation. Task-based measures, I think, are, I would say now standard in a standard way used in translational settings. Much less in large multi-site trials, but they're growingly used, and I think not necessarily as primary or secondary endpoints, but really to generate supportive evidence and to explore potential subgroups of responders or other things. That's at least been our approach, and the idea is we want to learn, even in case our current trial fails, we've learned something we can take into the next study and the next project. Many tasks have a little bit more of biological background to them because they've been developed as tasks for certain circuit engagement, and that certainly are the tasks that one prefers to use in an early translational setting, where you ideally would have the task that is linked to a circuit that is targeted by the tested intervention. And I think the fast fail trials that were set up a couple of years ago also had this type of philosophy or approach. But in reality, at least for us, we understand that we're exploring broadly here, and that it's not always a very focused or scientifically validated thing to do it, but we feel you have to start somewhere. Because there is a huge gap in clinical experience, and there is some guidance and information transfer from these deep phenotyping studies, BSNP, PRISM, and I'm sure from EMBARK as well. And that will help, and I think even advance further utility for many of these tasks. If you sit in a pharmaceutical company and you're designing a $20 million plus study, you sort of have a lot of pragmatic considerations, and anything you add to the study will add costs. And so aspects of accessibility, standardization, multi-site deployment, all these have an impact on the budget for the study. But not only for the study, unfortunately, also for the investigators. We have studies ongoing now, I have to say with some shame, where an investigator has to access 7 different portals in order to get his data through. Because you have your lab vendor, you have your ECG vendor, you have your IVRS randomization system, you have our electronic data capture, and then you would have whatever portal you use for the task to promote those data. And that adds to burden on sites, and burden on sites translates into recruitment challenges and issues in performance. I want to do a plug for digital technology as a huge opportunity, and I mentioned it, and I think we touched on computational psychiatry and all the machine learning stuff that you can do. And it's highly complex and not so easy to understand. I don't think we figured out, at least from industry side yet, how we may want to use it, but I think we're very open to look into it. And so in conclusion, I definitely would confirm that there's a growing interest and actually also use of task-based measures in translational and early stage studies, in particular for internal decision making. And that means the release of next stage investments. There is exploratory use in multi-site studies, and we expect these to guide our further use and a decision whether we want to go this route of formal qualification and in which way. And so finally, I think this is an excellent space for pre-competitive collaboration, because taking some of these tasks and making them drug trial ready, that's a challenge. There is the obvious lack on validation studies, and I think we touched about many of these aspects. And then definitely, I think the industry has completely moved from the way we were when I started. So we're very open to data sharing. And our placebo-controlled trials inherently have a test-retest arm in there. So I think it's a great opportunity if one can do it at low tech, low costs, and include some of the stuff in the studies. And with that, I'm just going to leave you with a couple of links for additional information, and I'm open to questions.
Are there any clarifying questions from the floor? So a quick question on the digital biomarker. What are your thoughts in terms of being able to use, because it's a measure of like real world behavior, right? Being able to use that as an outcome measure that may be more reflective than, symptom scale, especially for SMI type populations, in terms of both the whether you can use it and how you would deal with FDA.
So I will let the FDA speak to that if they want to respond to that. But from our perspective, we don't want to change the world and go for the Holy Grail immediately. As I mentioned, I think passive monitoring, or even if you interject active questions, they're already a huge improvement compared to the diaries we used to have. And in that sense, if I have a functional endpoint and I can say, well, we've improved 3 points on this scale that assesses activities of daily living. But I can also show you that these people now walk around more, they call more, they receive more text messages, they send more. I think that makes a pretty good case. And I think at the moment that for me would be sufficient. Whether I would really want to go the stretch and then say, let's make this a really secondary endpoint, I think that's maybe out there. But for us at the moment, I think if we can corroborate the evidence we generate with the established and accepted validated scales, I think that's already a big one.
Bill said I have this much space, so I will use it. The reward learning approaches and the reverse probabilistic reversal learning approaches really came to us from animal studies. So this seems like these are ideal behavioral targets for doing translational studies of drug effects on behavior. Is that working its way into industry to develop these behavioral approaches as translational strategies?
Yes.
Yes, thank you. Continuing on this and maybe following up on Dr. Pollentier's brief comment about maybe some limitations of the matrix or implied limitations. Our next speaker is Raquel, who is at the University of Pennsylvania and has headed up a very important cohort study of development. But more broadly, she and her husband, Boehringer, have been very involved in thinking about implementable tools for cognitive measures across disorders and the lifespan.
Thank you very much. Yes, so while waiting, I will say that this has been highly informative to go across disorders and see some of the doors in a way that have been opened. And in the last today more, we've heard more about the challenges in psychiatric disorders. So I say don't despair. The dementia people have come a long way, and our patients, those with psychosis and other severe psychiatric disorders, start at a very young age. So imagine that you're a parent, and this is something that I see frequently. You're a parent, and your child made it to Penn, a competitive school. And during the first few weeks, the behavior changes. And it's not drugs. And they're coming to the counseling center, and automatically we are called because the behavior is bizarre and it looks like they might be psychotic. And the parents were highly educated, doing great, fly in, and they see me. And when I tell them that I'm not yet sure, but it seems like this is what might be going on, inevitably, the mother cries and says, I wish you'd have told me that my child has cancer. There's more we can do nowadays for cancer than we can do for psychotic illness. So I personally do not feel it's a choice. With what we have to do the best we can to offer more to thousands and thousands of patients and family globally. So with this in mind, I'll tell you what we are trying to do, and I will point out, the slides can be busy, I will point out the take-home lesson. So when we traverse the line that we are over discussing from basic to translational, clinical, trying to go to populations with better therapeutics that will alleviate deficits, clearly there are genetic underpinning genetics. On some of them we know more, on some of them we know less, that have to be integrated. We have made progress in schizophrenia with the Psychiatric Genetic Consortium. You'll hear more about it later. Soon, we are talking about 250 loci with a polygenic risk score. We have to tuck it on the side because it gives us an opportunity to parse heterogeneity, and it's so complex that we need everything to bring together. Environment, when I say environment, I will illustrate it for people at risk for psychosis from intrauterine environment. Environment matters. So we need to be able to measure it, and we can. Both impact the brain, and the product of the brain are behavior. So we see neurobehavioral deficits across psychiatric disorders that have been nicely illustrated today, and we see the symptoms, an array of symptoms that are very heterogeneous. Is one of the things that people ask me, how long have you been doing schizophrenia? Not two people are alike. You know, there's marked heterogeneity, just the way we heard today is for autism, for Alzheimer's, for Parkinson, etc. So, can we parse the neurobehavioral domains that can be measured with multiple tests? Can we reach some agreement that there are certain domains of behavior that can be clearly related to underlying circuitry in human and in animal studies as well? We can, and I will illustrate our executive function we heard about, memory, complex cognition, social cognition, reward and motivation we just heard about, and sensory motor function that is important. The domains of what I will call what is evident in psychiatric disorders, broadly termed psychopathology, pathological behavior, that's how patients present, relate to anxiety and depression, fear, externalized behavior, and psychosis. Both of them impact functional outcome. And our goal is to come up with reliable, rigorously with measures that can be applied in populations and we can intervene and come up with a better outcome. So the study of neurobehavior in psychiatry, neurobehavioral deficit, I mean behavior that can be linked to brain circuitry. The term is neurobehavior here defined. Thus, they represent brain dysfunction. They're evident across psychiatric disorders, perhaps more so in schizophrenia than in others. They impact functional outcome. The deficits in cognition are evident before the presentation, before they meet criteria. So it's an early sign that something in the brain is happening, that the process is underway. Integrated with neuroimaging that we heard about, with neurophysiology that we heard about, they enable a mechanistic probing of the underlying circuitry. So if we have to tweak the brain, we know where we are going. That's the goal. So we can impact a specific domain that is associated with major deficit and is relevant for functional outcome. So it can provide both biomarker for stratifying patients, and I will illustrate they're not all alike. One of the difficulties that were in multi-site study for cognitive, and I participated in order to learn and give our patient a chance, my gosh, some of them were 2-3 standard deviation from the mean, and you want to bring them to normal? There's so much heterogeneity, and it depends on how focal is the deficits and how diffuse. This needs to be assessed, and the outcome measures that can be target for intervention. And that's the uniqueness of some of the quantitative measures. So I will illustrate the data with effort that we have done, others have done other efforts. I'm closest to our data that I was asked to talk about, of the PEN computerized neurocognitive battery. So it was developed in order to use it as a biomarker integrated with clinical characteristic in order to enrich the clinical trials that focus on cognitive performance that is related to brain system. It provides measures on 12 neurocognitive domains, both performance on quality, accuracy of performance and speed of performance. There's standardized acquisition. People can be easily trained to administer the battery. Can be done in person, can be done online. They can get a certificate, and you can follow and see if there's any drift. Every coordinator who does it is getting a certificate, and it has been applied throughout the world. So some of the sites in South Africa, for example, in South America, people who have never touched a computer are trained to do it, and to do it extremely, extremely well. Translated to 15 languages with data on controls and on patients with a very similar structure across sites. All the QC is online and uploaded. There's no human interaction. The algorithm for scoring, and you get an immediate feedback when you're done doing it. It's 24 hours, 7 days a week. You get a feedback of something that you've missed or something that is wrong. Takes about an hour. These are just examples. What we had in mind is a lifespan perspective. We wanted to start with young children, the precursor to developing psychosis, for example. ADHD might be one of them, same frontal dorsolateral prefrontal circuitry. We wanted to be able to do it with kids and to do it also with older adults. And with each of the domains, there are multiple stimuli that are presented, and it's up, the pace is up to the research participant. People with significant deficit, such as rare CMVs, 22q11.2, with proper instructions, can do it. So before rolling it out, we validated it in functional MRI. So the entire task by task we built to show that it recruits a unique circuitry. Then we put an entire battery, 1 hour, in the fMRI for healthy, typically developing but healthy young adults, and were able to show that what we have done in task by task now applies across the battery. So we can tie to underlying circuitry. It has been done for some of the participants with EEG, with multiple electrodes, but all have undergone the fMRI study. The importance of it is that I wish we can put everybody in an MRI scanner. We can't. So at some sites, especially remote sites, that some of the studies are conducted, you know where you are as far as circuitry based on the performance. So this gave us an opportunity when we were done with that to do the Philadelphia Neurodevelopmental Court, a word about it, recruited from Children's Hospital, pediatric, non-psychiatry kids. They were genotyped before. We have access to electronic medical records, very diverse population, age 8 to 21. So there were close to 10,000 kids. It's all in the public domain. And this is what the kids have undergone. They've undergone with trained clinical coordinator, a structured, all computerized, except for assent, consent, nothing is on paper. So the undergrad is structured evaluation, that is an adaptation of the key decides, a structured interview that, every journal is happy about and study sections are happy about too. It's very structured, and you do everybody exactly the same. There was collateral information from the mother, commonly, sometimes the dad next door. They all underwent in the middle the computerized battery, about an hour, randomly selected 1,600 undergone multimodal imaging. I'm going just to talk about the cognitive today, but it's all related. The imaging is related to the cognitive. Okay. There's multiple items, multiple domains. Can we reduce them? Absolutely, reliably. In all the studies, we get studies from all the data that other people are using the battery. It's free, it's in the public domain. Many people use it, it's their data. But for the normative data, they want to check if the structure is exactly the same, the factors, and I can tell you that it's identical. So we get the factor of memory, complex cognition, executive function, and social cognition or emotion processing. They're related? Yes, but you can pick one factor. If you have a target that relates to executive function, you can pick up the task that relates to executive function. So it's flexible, it's modular. The clinical assessment also yields several factors, and this was a bifactor model. There's fear, and that's where I took the terms from. It's empirically based. Anxious misery, or it's anxious anxiety depression, many times correlated, especially in young people, but also in adults. Psychosis, which is what is driving us, and externalizing behavior. And this also, for those who have used it in other populations, emerges the same factor structure. we have a ton of normative data. So these are people that have the stamp of typically developing based on clinical presentation and evaluation, electronic medical record. So we have norms for males, females, degree of education, ethnicity, you name it. And you can see that we have begun to go into older adults as well, although the majority are age 8 to about 30. And this is, this is the typical time, more in the late adolescent, early adulthood, that psychosis emerges. So the properties, in order to evaluate its suitability, if it's ready to prime time, we looked at this test, retest, reliability, stability, assessed in different culture, translated with different languages. And some of them are a week apart, some of them are 16 times administered. It went to space with the astronauts, so they had repeated measures with the twins, so we know quite a bit of what time of day you do it, etc. So all of this is either published or on the way to publication. Sensitivity and specificity, there's good factorial structure that I saw that is common across the site, and we've applied it, and I will illustrate to clinical high-risk people for schizophrenia. I mentioned it is heritable, and I will show you that. I mentioned it in the public domain. It's modular, and it's currently began to undergo adaptive, computerized, look at adaptive testing using IRT so that we can cut it to about less than half an hour from 1 hour with multiple forms, so there's lack of practice effect. This is just to show how we established reliability. This is an NIMH-supported study on multi-generational individuals with 2 first-degree relatives having schizophrenia. So it's multiplex families. And what you can see, the red line are the person with schizophrenia, the proband in the study. And you can calculate efficiency. Accuracy over speed is on the left. In the middle, you just have accuracy, and you have speed on the right. And what you can see when you look at efficiency, one score, the red are people with schizophrenia, they are most impaired. The line above them in orange are first-degree relative, less impaired. Second-degree and third-degree relatives are the yellow line. The blue line, 0, these are standard deviations. You can see that the degree of proximity, familial, genetic proximity, is related to the degree of impairment, both more for accuracy than for speed. This is another study, COGS, that Greg referred to, and this provided us with an opportunity to administer it in 2 ways. One, people, as part of a family study, quads, a patient with schizophrenia, and 3, 2 parents and a sibling without schizophrenia, was study 1. And again, the 0 line are normative controls. And what you can see is that if you look at accuracy and speed, is that people with schizophrenia, the red line, are more impaired compared to controls, and they are slower. Note the yellow line. The yellow line is the second study. It's not a family study. It's individual with schizophrenia, case control design. When parents are not involved, just the individual comes, they're more impaired. So just by the simple design of the study, is it a family study? Is it case control? I can tell you what that they are going to have more cognitive deficit and will be harder to treat. Okay, that's a third study, partners, African American, 8-site study in the United States, supported by NIMH. If you just look to the right, you can see this is irritability. It's a family study. The red is accuracy. The blue is speed. It's irritability. Accuracy is more heritable than speed. So this is now, I've shown you both for multiplex families with schizophrenia, the COGS family, case control, partners, similar degree of irritability across domains and populations. So in longitudinal CHR studies, we wanted to examine if the performance relates to the clinical characteristics and if we can stratify for future studies individuals by the cognitive presentation or performance on this battery, and it can be applied, can we apply it as a biomarker that can be translated to brain systems and evaluate the outcome measures as target for intervention. So we looked primarily at efficiency and standard z-scores across domains. From the 10,000 people, about 10%, remember they came from pediatrics, they're not help-seeking, they're screened. 10% of them had subthreshold psychotic symptoms, significant enough, mean age 15 of the sample. Significant enough that they are causing them distress, declining social functioning, and impairment in school performance. So it's a student who went from A to B, from B to C, etc. By their report and parents, collateral information. And what you can see if you look at accuracy, PS, psychosis spectrum, subthreshold psychotic symptoms. You can see that across the board, they are impaired already. Neuroleptics, psychoactive medications have not touched their lips. Okay? So they've never been hospitalized in this subsample. So they just have subthreshold psychotic symptoms. Otherwise, they're typically developing. We excluded those with neurological. So these are healthy, moving, behaving kids with subthreshold psychotic symptoms going around with them. Language and some of the social cognition deficits are most impaired, especially in speed. If you see the two dips, it's in language and in social cognition. It afforded us the opportunity to look at the course of symptoms in relationship to neurocognition. They were followed at 2 years intervals. Because they are young, psychosis the risk period is not over. On the left, you can see the neurocognitive domains. Blue, TD, typically developing. They came in as typically developing, and at follow-up remain typically developing. No subthreshold psychotic symptoms. Healthy kids, I call them the speak and span kids. Everything is going great. On the orange line before are kids that at time 1 came in and they were typically developing. Within 2 years, psychosis emerged. Remember, they're young. We went back and said, at time 1, in what way did they differ from the follow-up period? What distinguished them? If I put differently, if I would have had the measure at time 1, would this kid be at higher risk, considered without symptoms? And what distinguished them is the cognitive. So if you look at the cognitive domain, you can see for executive function, the one of the left, especially for working memory, they are impaired. They are impaired for, less impaired for the memory component and somewhat impaired for the complex cognition and social cognition. So at time 1, without symptoms, we can see that the cognitive measures are sensitive to those who emerge. Let's reverse it. Those who came in as typically as TD, as the subthreshold psychotic symptoms, again, the red line, and at follow-up persists. Some of them, many of them have gotten worse, and some of them, about 8%, now meet criteria for schizophrenia. Commonly, they're a little bit older. They're still adolescent, but a little bit older. Now look at those who come in and they're resilient. They don't have psychotic symptoms anymore. Possible? Yes. People can have report with stress or a subthreshold. And there are many people who walk around like that and they don't have schizophrenia, fortunately. But if you look at the cognitive, you can see at intake that the resilient kids with the subthreshold psychotic symptoms did not proceed, did not progress, at intake already had better neurocognitive performance compared to their persisters. And if on this basis I will say, what do I want to target? You can see that working memory is important, language is important, and some of the domain that relate to social functioning. For working memory is translatable. If you think about the D1 partial agonist that has been shown in non-human primate for processing spatial working memory task, can we target it in people who are on the verge of psychosis before they have the break, before the train derails? with a very targeted approach for working memory. Perhaps we can, we haven't tried it, but this is the level of pushing forward with data that is accumulating and replicates. I said environment matter, and it's something that many times in drug trials we don't consider. I think it's important for stratification as well. We can't assume that when they come in, there's a path to every person, and this is just looking at stress load, traumatic events in the risk calculator that has been published by Ty Cannon and the Naples group is important for transition to psychosis. We see it as an important factor in our sample as well. That those who transition have more of a load of traumatic event. Traumatic event impact cognition, and you can see that some areas are more sensitive. This is the load of traumatic event. I mean, traumatic events, it's not like I didn't get what I wanted. It's it's somebody pointing a gun at you, it's seeing somebody hurt, it's being sexually assaulted, real traumatic event, okay? So from 0 on the left to more than 3 on the right, you can see that some domains, not episodic memory, but executive function, reasoning, social cognition, are related to traumatic event. And these are people who don't have PTSD. And now if you look at the environment, another domain, and that's important as my experience is that many of the people who participate in treatment studies are people who are coming, at least in the United States, from lower socioeconomic strata. We need to think about that, and try to get people to understand that if it's a brain disorder, you are part of you have an obligation to contribute to research. So for example, these are people from adverse poverty, from lower socioeconomic status. And what you see that in cognition, they're poorer to begin with. This is the total normative sample. And now if you look on the right, you see that traumatic events do not affect as much the effect size that relates to cognition. You see it in complex cognition, you see it a little bit in the executive function, but it dwarfs compared to the socioeconomic. So stratifying based on that can be very helpful in achieving targets, and parsing the samples for that is important. Connecting it to polygenic risk score, it's an example of collaboration with the Broad. We looked at the schizophrenia polygenic risk score for the entire sample, and what you can see is that there are 2 domains that jump out. One is related to verbal reasoning, the other is to emotion recognition, the 2 red lines that pass significant, and data sharing, it's related to, below is the polygenic risk score from lowest quartile to the 4th quartile, from the lowest to the top, and what you can see is that greater deficit is for those who have a higher polygenic risk score for schizophrenia. Randy Buckner, we shared the stimuli with him in a totally independent sample for the emotion, got exactly the same. So it's an opportunity to integrate the polygenic risk score and see what is our target. And I bet Leah will talk more about that. So the summary of direction is that the neurobehavioral measures can provide really a critical and needed bridge between the mechanism, because we have the circuitry that underlying it, many from some of the domain also with animal studies, and they offer both a biomarker and can be an outcome measure. They can become a pivotal component for neuroscience-based intervention. So we go well beyond the standard, both in the evaluation of the patients and in the stratification. It's feasible in large-scale studies. I haven't seen a participant yet who came to a study and didn't want to do it. They love interacting with computers, and they do it really well. And I was surprised to see the quality of data that comes across from remote places, small villages in South Africa. Amazing. You train them, and they're all on board. So it's exciting to see that. Longitudinal studies can be easily done for stratifying before and for following up the patients. Repeatable measures are available. The variability in performance, in informative, there's high fidelity, can be translated, as I said, to brain systems. It's heritable, related to polygenic risk score, and it allows cross-syndrome dimensional characterization. So, for example, we see that patients with schizophrenia are different from those with major depression who get the same battery. As I said before, we are currently at the phase of integrating it with advanced psychometrics so we can make it short and it's modular, and new modules can be added, like the reward module. We are using the balloon test that you were mentioning was added. It depends on the investigator and what are the hypotheses for the study. And these are the people who persevered throughout the time, and others have come and gone. All have contributed to that, were supported by NIMH. Thank you.
Our next speaker is Laurel Beckett, who's going to bring us back to a consideration of trying to use more than one measure. For those of you who don't realize what Laurel has done, Laurel has been the lead statistician for the whole ADNI effort. So she's been, over the years, what are we now, 15 years into this?
Something like that, yes.
Struggling with the issue of how do we combine measures and get more information. Laurel?
Thank you very much, Bill, and thanks for inviting me. This has been a really impressive conference so far, a symposium, and I hope that this will not be the first and only. I hope there will be more. This is a lot of learning going on. First, I want to apologize. I know I'm in the session on cognitive measures, and I'm actually going to use, or clinical measures, I'm going to use more imaging things to illustrate. And the second thing is, this is really going to be a little more about methods, but I promise you we don't have equations, so don't get up and run away. There we go. So I don't have any conflicts to disclose, but I do have thank yous to especially Danielle Harvey, who's been with ADNI since the beginning also, and Naomi Saito, who's our master's level statistician. They are terrific colleagues. I'm very honored to work with them. So, overview. I am going to be talking about biomarkers for prognosis and prediction. So I know yesterday we had an introduction to the classes of biomarkers. The things I'm going to talk about will be applicable to other classes, but that's what I'll use to illustrate. And I'm going to start with the example of hippocampal volume, which was qualified as a biomarker through the EMA, and Diane Stevens and I have worked together on this with many other people. And I'm going to just sort of show a little bit of the criteria for qualification and some of the limitations of using one marker. Talk about some strategies that you might use to try to bring multiple biomarkers together, and the sort of two common things that we'll illustrate are creating a score or a one-number summary and putting together something more exotic or elaborate, partitioning the space. So think of the multi-dimensional space of all the possible outcomes. And actually, we had a little glimpse of this yesterday from Cliff Jack's categories, and then labeling them in different ways. So that's sort of like building a tree. I'll take a look at some potential biomarkers and how to compare them. So if you add something on top, so you take hippocampal volume and pile more things on top, how can you compare those? And again, context matters. And how do you compare two sets of markers or combinations that were not built by piling something on top, that are sort of different strategies? And again, anything we do should allow for proper statistical inference. Okay, let's see if this is, okay, so the treatment goal here that I'm going to talk about as an example is preventing progression from mild cognitive impairment to AD. So I've worked in other areas and hang out in other parts of the neuroscience space, but as an ADNI person, that's handy data and handy to think about. So many people, maybe even most, with MCI will progress to AD, but not all. So there's heterogeneity that can be a challenge. Our goal, though, is to figure out who you would like to prevent progressing and then prevent it before before the brain is irreversibly damaged, at least with current treatment options. And There may also be issues that the heterogeneity is not just in whether people progress or not, but there may be need for different targets because people have different things going on. Although, speaking as someone whose parents lived to be 100 or 101, you know, you get old and there's just crud in your brain. The technical term, no. Anyway, for some people, my dad was 101 and pretty sharp, but so we have some potential roles here for biomarkers in this context. And again, context is important. So if we want to talk about identifying people most likely to progress and targeting those people, that's a prognostic biomarker. If we want to talk about identifying those most likely to respond to a targeted therapy, that's a predictive biomarker. So hippocampal volume actually has already qualified as a prognostic biomarker for MCI in Europe. And this is CAMD. Diane, big cheer for Diane's hard work in leading this. The evidence, it was substantial that's required for this, and our FDA colleagues will know this as those of you who've worked on such biomarkers. So there's a lot of evidence out there in the literature already that it is conceptually plausible because cortical atrophy is a part of the basic definition going back to Alois Alzheimer. The performance is very reproducible. It's reproducible in the short term in the same people, and we've done that in ADNI, other places also. It's reproducible across instruments. It's reproducible across labs, evaluating, it's calculating. It's reproducible across algorithms. Pretty much, you know, if you have a change in one person over several years, it's probably real. And if the person looks stable, that's probably real. So that's encouraging. It also correlates with measures of diagnosis cross-sectionally. And the baseline is predictive of change in whether you think of it as progression in disease category or change in some cognitive function or behavioral measure. And we replicated these analyses in multiple data sets, including ADNI, and found indeed it's a predictor of progression. The area under the ROC curve ranged from 0.73 to 0.76. And we then further looked at scenarios for use in actual clinical trials. And I want to emphasize this, if you're trying to say this is the context or purpose of use, it's really important to look at this. And in fact, if you use this for screening, you can have a 35% increase in the 2-year progression rate and a corresponding increase in power or decrease in sample size. Now, of course, you have to screen people to get them there, and that may be an issue too, and we'll see a little bit about that. But it's not perfect, right? That ROC curve isn't perfect. Maybe we could do better than the 35%. Okay, how do we do this? There are candidates that we could use to improve, and we could use, you know, cognitive function or various things, but I'm going to illustrate with two relatively simple things, just as a sort of basic example here. Genotype, how many, whether people have one or more copies of the apolipoprotein E4, and evidence of whether you have amyloid in your brain. And we're using, since we have ADNI, most people in ADNI in this phase did in fact get amyloid PET imaging, but a few of them didn't. So for those people, we looked at CSF, and we required that everyone have all 3 of those in this little analysis. They're all MCI to start with, and they all had to have 2 years of follow-up because my outcome was whether they progressed in 2 years. That led to 269 participants. And being a good statistician, for this simple analysis, we split this into a training set and a test set. It's really important that we develop the models in the same training set and compare them on the same test set. Okay? So, 2 strategies. One strategy is to construct this common score, you know, some summary weighted measure of some sort, you know, weighted scale. And depending on the outcome you have, that's usually done with a regression model. So we have progression, yes, no. So for that, one typically uses logistic regression. There are other things you can do, and that's what we used. If we had looked at time to progression, we could have used a survival analysis. If we had looked at rate of change in ADAS-Cog or something, that could have led to a mixed model repeated measures. And in all of these cases, you get like a single number summary that's a weighted combination of the different markers. More elaborate things could be constructed using, if you want to be really fancy, some kind of exotic machine learning. We used a fairly simple classification tree to illustrate because there are only 3 variables. Okay, logistic regression. So I'm going to start with just the hippocampal volume as kind of a reference to see if we can do better. And in this little data set, when we evaluated our proposed score in the 134 people test set, the odds went up 4-fold for every 1 cc decrease in hippocampal volume. And the ROC curve, you can see there, and it had the area under the curve of 0.709. This is not too bad, I guess. And kind of in the ballpark of what we saw with other data sets. Okay, can we do better? Can we add 2 more variables and do better? And in fact, adding 2 variables, we find that hippocampal volume still predicts. It's a little less striking. It's a 3.3-fold increase. And amyloid positive predicts pretty strongly, 12-fold increase in your odds of progression. E4 doesn't add a whole lot if you know those 2. They add if you know the others, but this one doesn't add. And the ROC curve is a little better, right? It's 0.78. Is 0.78 bigger than 0.709? Yes, no? Votes? Maybe. We've got a winner. Maybe. So here's the deal. If you want to compare those, you need to do it properly. Okay? They were based on the same 134 people. So you really need to line the people up, like 134 people in a little row, and see which one classified more accurately in the ROC curve land. Was the ROC curve closer to the truth and there's a lot of ways to do that. There's a pile of literature. People write dissertations on it and stuff. We used a bootstrap approach and found, the probability we'd get a difference between them that big by chance alone was about 0.095. And it's pretty similar for other approaches to testing. So this difference could be just chance in these 134 people. Okay. Now, the other thing that people often do is say, well, did the logistic regression fit better? And you can test this too. And in fact, we added 2 things. So we started with 1 variable, added 2 more, and you can do a 2 degree of freedom. This is not a coincidence, it's 2. And that test gives a p-value of 0.005. So the logistic regression is better with 3 than with 1. Okay, anybody want to think about that for a minute? It turns out it matters what question you ask, whether something's a better predictor or not. So is it better with 3 variables or not? Well, maybe neither one of these is really the question I should be asking, because I started with a context of use. So what I really want to do here is not just say, do I explain more and how am I going to calculate explaining more? I really want to know, clinical impact. Does this change my clinical trial design? Am I going to get better clinical trials? And, power of a clinical trial actually depends on the number of positive out of the people at least in this kind of design, where I'm looking to see if they progress in 2 years and if I can cut that down. So suppose I have a little standard design. This is the one we always use for evaluating ADNI markers. I say, okay, I want to reduce the progression rate by 25%, 80% power, alpha of 0.05 two-sided. I'm tired of that little design. We use it all the time. But it's kind of the standard thing. A screen that's more stringent will make a tighter, more specific group that has more people in it, higher proportion who progress, and it will have greater power. The price you pay is it's less sensitive. It's going to miss more potential participants. So let's see what the numbers turn out to be for this. In our test set, 25 of the 134 patients progressed in 2 years. That's 19%. And if I were to do my magic study design on those 134, and data based on those 134, I'd require 1,892 patients. Big study, right? But if I were to say, hey, I've got this cool one thing, hippocampal volume, it's been approved, let's screen on that and use a kind of standard screen that we used in our in our design, I have 40 people out of the 134 identified as high risk, 16 progressed, that's 40%. And if I were trying to reduce that 40% progression rate to 30%, 25% reduction, I could get by with 752 people. So at the price of having to screen more to get up to 752, maybe, I could have a better, more powerful trial. Interestingly, my 3-variable thing also yielded 40%, 18 out of 45. No difference at all in their impact, except that maybe I would get a few more people with my 3-variable guy. So these formal comparisons are useful, but you have to, again, come back and say, how much would it cost to screen more people? If it's a cheap screen, it's probably worth it to get by with a much smaller sample size, more efficient sample size, right? Okay, let's try one more approach. Let's try a little classification tree. And I did a real simple tree here, and it actually came out very nicely. Said, okay, first question is, are you amyloid positive? If you're not, poof, you're out. I'm not interested in you. And only 4 of the people who fell out there in the little blue box on the left were positive, progressed. If you are amyloid positive, then we look at hippocampal volume, and the magic cut point is 3,064 cubic millimeters. And that yields 10 of 48 that we miss, but 11 of 19, 58% that my screen identifies. So it reduces it down to 19 people, but well over half of them progress. So this is my 3-variable little tree. And it's really good on specificity. Doesn't have very many false positives, but it has a lot lower yield. It's missing a bunch of people who do progress. So a formal comparison of that is also appropriate. And the formal comparison we do is we separate sensitivity and specificity. We look at the true positives, the true progressed, 25 of them, and we find that the tree and the logistic regression model both got 7 of them right who didn't progress. They got 11 of them right who did. And the tree missed 7 people, left them out, who progressed. So it's significantly less sensitive, McNemar's test. And of the 109 who did not progress that I wanted to screen out, both the model and the tree screened out correctly 82. Both of them let 8 slip in that they shouldn't. But the tree kept out 19 more who it shouldn't. So it's more specific. Now, I could tweak the parameters and make it a little different. And that's something that you want to think about, right? If you're designing a model, is what are your costs? Does it cost more to miss people you should include or to include people that you shouldn't? So that is the basic approach. And I think that this applies to all these wonderful examples we've seen. I think our previous talk had beautiful validation. Thank you, Dr. Gurr. That was a very nice example of all these different ways of validating your markers. And when you start thinking about clinical trials, again, critical to look at them in the same people. You have great large samples. You can do all kind of cross-validation. This is terrific. But the take-home message is, first of all, is if you just have a few predictors, not like, a bazillion items or proteomics or whatever, just split them into training and test sets. And if you want to be fancy, use the K-fold cross-validation. That's okay. And make sure that every potential candidate is evaluated on the same test sets. If you have lots of potential candidate markers, then you may want to do a more exotic thing. The sort of best practice seems to be have one set for picking your predictors, the next data set for building your model, and the third subset of the data for testing your predictors. And again, this should all be done so that everybody gets compared on the same people. Okay, Everything that you do, that you validate, should be accompanied by proper things like robust 95% confidence intervals, and every comparison needs to be properly accounting for the fact that if you're doing it right, you're comparing in the same people. And I realize that harmonization is a challenge when you have multiple data sets, but if you can manage this, this is really important. And the tests that you use should take account of this. They're block tests, like paired t-test. We showed McNamar's test, and evil Microsoft turned it in here into McNamara. I don't know why. It doesn't like McNamar. This is Microsoft thwarting me. That's why I use LaTeX and left to my own devices. If you're going to be doing this for FDA validation, obviously there's a lot more to do, including multiple data sets and harmonization and many, many, many other challenges. But the core principle I want to establish is that there is a proper statistical technique for doing this with care. And it's a little frustrating. I know we mentioned yesterday we had questions about what about people who use ADNI data. And if you look, there are several thousand papers that use ADNI data. And some of them say, I have the best combination of markers to distinguish or predict, and they don't do it right. But as I said yesterday in my comments, I can't tell everybody to do it right, but I can tell you guys. So I really appreciate the chance to be here. And I want to note also that all of these principles apply to other things. ADNI's not yet at the stage where we can do surrogate markers. Sadly, we don't have a treatment that works in order to say that the surrogate catches the effect of the treatment. Even when you think you have them, I worked in AIDS trials, and it didn't always work the way we hoped. And the previous attempts that said, a likely surrogate, we think this marker is going to predict how people will respond to this treatment. Our phase 3 trials have not actually borne this out in a lot of cases. So surrogate markers were not ready yet, but we can use them for prediction. We're trying that. We're hoping that we will be able to identify people who will respond to targeted treatments. If you have an effective predictive marker, suppose, for example, we have something that's really effective in amyloid positive people, and we use our little amyloid positive screen, our little 3 guy. If we could reduce from 40% to 20% instead of to 30%, we could get down to a study of 182 people. So predictive screening, targeted, personalized therapy is very effective. So that's the direction we're hoping to move. We're all trying this with biomarkers. ADNI is, one of many. I think that having made ADNI data public has been a huge help in our field. It hasn't generated treatments yet that work, but trying things out in publicly available data is critical. So again, I'd like to thank you all for the opportunity to be here, and this is a fascinating study. I knew it was going to be. Everybody's looking very attentive. So if you have questions, I'm happy to answer questions. You always do. I love that you always have a good question.
It's just something that built into your equation, and it was really sort of a cost-benefit analysis of screening versus. But do we know yet whether we encounter in Alzheimer's trials what we encounter in depression trials? That is, that you actually lose signal detection sometimes by increasing sample size because of uncontrollable increases in variance. So empirically, there is an argument to be said of not allowing your arms to get too big, because implementing complex multi-site trials, your variance of those trials is totally unpredictable in many areas. I don't know if this is true in Alzheimer's, but it certainly was true in depression. And it may be, Stephane, maybe others can be, it may turn out to be true in the schizophrenia, the way we're doing them. In other words, you can't really predict what your variance is going to be.
That's a great point. And while I don't know what's happened in trials specifically, but I know in our observational studies in Alzheimer's that we see considerably more heterogeneity. So ADNI tried to be really specific and get people who looked Alzheimer's-ish, memory impaired, blah, blah, blah. And we still had heterogeneity at postmortem. Community-based studies like the Chicago Healthy Aging Project or like our work in the UC Davis find substantially more of other things. And if you so our study, we have a paper that's just about to come out in Davis, that there's a lot of vascular damage. And if you look at our Hispanic cohort, very little pure Alzheimer's at postmortem. There's a lot of vascular combined with Alzheimer's or vascular by itself. And one would expect that would lose signal if you're targeted treatment. So, but on the other hand, if you're trying to study what will work in the population and you have 10% of people 65 and older sooner or later developing dementia, at least we have to study people and then subset, stratify, target. At least, so that's. I hope that does that help.
I just wanted to highlight a point.
It's an important point. It's an important point, and in cancer we've gotten down to N of 1 trials.
Yeah,
so
that's a little bit. Thank you very much, and we'll take a quick break. I was just saying we need a larger sample size for genetics talk in the audience here. Okay, we're getting there. The last talk in this session is lead efforts from the University of Vanderbilt, and we all want to know what we might do with polygenic risk scores in these very complex areas. I leave it to you.
Okay. Well, thank you so much for inviting me to speak. It's been an incredibly educational experience for me because there really is a lot of excitement about the use of genetic risk scores as potential biomarkers. But at least, I can speak for myself as a geneticist, really the framework for biomarkers and their evaluation and their use is pretty foreign to me. So I've learned a ton being here, and I hope in the last 20 minutes or so of the session I can be as engaging as the other speakers and keep your attention for this talk as well. So there was a mention earlier, I think yesterday, somebody mentioned the kinds of peaks and valleys that we have in terms of enthusiasm for different biomarkers, and certainly genetics is not immune to this. And so it really is a pivot to be here talking about some realistic possible use cases for genetic markers when just a few years ago, the popular media was reporting that genetics had failed. And so I'd like to kind of make a plug for trying to keep a steadier course in terms of our expectations of what genetics can do and our willingness to kind of give it a try. So with that, I'm going to talk about polygenic risk scores for patient stratification and biomarker development, and I'm going to give some examples of work that we've been doing in a healthcare system. So I have no conflicts of interest. And I just wanted to start off by reminding everybody that that really the idea behind using genetics as biomarkers is rooted in the observation that all psychiatric disorders are heritable. All psychiatric disorders, the phenotypic variance within the population for psychiatric diagnoses is influenced by genetic variation within a population. And so what you're looking at here is a plot from a paper published a few years ago. On the y-axis, we have the heritability estimates for 19 different disorders listed on the x-axis. And the light gray bars are the twin-based heritability estimates. And so you'll notice that pretty much for all of these neuropsych phenotypes, those heritability estimates range from about 0.3 to about 0.6, sometimes up to 0.8. The dark gray bars represent the proportion of genetic variance that is accounted for by, of phenotypic variance that is accounted for by common genetic variance. So that is the just the common additive contribution across the genome. And one thing that you'll notice is that those bars tend to be lower. So on average, for what we call the SNP-based heritability or the common additive component, we're accounting for about half of what we see in the twin-based heritability estimates. And that's an important point that I will come back to later on, but I just wanted to introduce it here. So in addition to the observation that all psychiatric disorders have a heritable component, and again, just to sort of refresh that vocabulary, when we talk about heritability, we're talking about the proportion of phenotypic variance in the population that's accounted for by genetic variation in the population. So again, all psychiatric disorders are heritable, right? So some of that phenotypic variance is explained by genetics. But even within that contribution of genetic variation, it's very complex. So we can have genetic variation that is very rare, so occurs very infrequently in the population, but has a pretty large effect size. So it matters a lot when that variation occurs. And so these are things like the 22q11 deletions and duplications, for example. And then on the other end of the spectrum, we can have common genetic variation, so variation that occurs all across the genome in everybody in the world. And the effect sizes for each one of those common variants is going to be very, very small. So generally a good bit less than 1.5. Odds ratios of 1.5. And we can have kind of everything in between. In addition, we may have genetic variation that contributes to risk, but really only in the context of another environmental risk factor. All right, so then there's another layer of kind of complexity there. And when we think about those gene by environment interactions, they may also be influenced by sex, by age, so we can get into mediating moderator kinds of models, for example. So a lot of complexity here when we're talking about just the genetic contribution, and that is part of that discrepancy between what we observe in our twin-based models, which include all of the potential genetic variation, and what we observe by just measuring the common genetic variation. So back to this, we should keep in mind that these dark bars here are really only reflecting this portion of the genetic variation. Now, within that portion of genetic variation, we have also learned that all of these traits really are very highly polygenic. So lots of risk variance distributed across the genome with very small effect sizes. And this means that we require massive sample sizes to be able to robustly and statistically significantly separate the signal from the noise in terms of a genome-wide association study. So basically for the last 10 years, the several consortia have been doing this, and really, again, drawing on some of the themes of the symposium so far, this consortium science is just absolutely has been essential to the successes of genetics in the psychiatric genetics field. So I have up here the Psychiatric Genomics Consortium, which now has, I think, over 1,200 investigators, hundreds of sites across the world, over 1 million subjects included. So it's a massive effort. The EyeSight Consortium, which is essentially studying the entire population of Denmark. I mean, really large-scale efforts. And data sharing is an essential part of the success of these efforts. So what I wanted to display with this figure here is you see on the y-axis the number of genome-wide significant associations that have emerged from each study. And on the x-axis, the number of thousands of cases that were required to reach that level of genome-wide significance. And so you can see a really big difference between, for example, IBD, which has a much different slope than height, for example. And schizophrenia and bipolar tend to track a little bit more with height. So this is actually demonstrating the extent of polygenicity that we're dealing with in terms of the psychiatric phenotypes. We we're hoping to get something closer to IBD, but that's just not how nature designed us. So the effect sizes are small, they're distributed, but they are detectable with large enough sample sizes. And so this is, again, kind of an example from schizophrenia and depression of the progress that we've made over the last decade in identifying with statistical rigor the individual genomic variants that are associated with each of these disorders. So you can see, for those who may not be familiar with looking at these kinds of plots, along the y-axis, we're looking at the minus log 10 of the p-value. Each dot is a genetic variant, a SNP. And along the x-axis, these SNPs are ordered by their chromosome. So we're looking across the genome. And you can see that in 2011, we had just a few genome-wide significant loci in schizophrenia. By 2014, the sample size was up to the tens of thousands, and we had, I think, 108, and now it's even higher, up to about 200. Same story for depression, and really it's been the same story for each psychiatric disorder. And so our hope was, I think manyfold, actually. We hoped that some of the genes that emerged from these genome-wide significant loci would be good targets for drugs. And actually that has turned out to be true for a number of disorders. But the other thing that has been really useful and important product from the genome-wide association study world is based on the notion that while GWAS is a great way to home in on those most significantly associated genes, really what we're doing is we're rank ordering all SNPs in the genome in terms of their association with a particular phenotype. So even those genetic variants, that fall below the genome-wide significance threshold level can be informative for example, for risk prediction. And that's one of the things that we're learning in the development of polygenic risk scores. So we all carry genetic risk for psychiatric disorders. And this is, I think, one of the fundamental take-home messages that has come out of psychiatric genetics. We all carry genetic risk for psychiatric disorders, and this risk can be quantified based on what we know about which variants are contributing to risk from those genome-wide association studies. And this can be also modeled as a continuous liability in the general population. And so our hope is that we can use this information to help with early identification of people who are at high risk, to help potentially with differential diagnosis, and maybe even to help prioritize treatment options. So this figure here is kind of a conceptual model of that distribution of genetic risk that then is also combined with environmental risk, other genetic risk factors, and eventually one gets pushed over a disease threshold. So I'm going to put some of the new vocabulary that I learned to the test. Hopefully I did this right. But I think that based on what we've discussed here today and yesterday, polygenic scores may best be characterized as multimodal, integrative, surrogate, and risk markers. And so multimodal because it's not one genetic marker. There are hundreds or thousands of markers that are being combined. Integrative because we are combining them into a single score. Surrogate because we're not measuring an actual outcome, and risk because we're using them to predict risk for either the disorder or potentially prognosis. So how do we actually measure polygenic risk scores? Well, there are actually a lot of ways to do this, but they all kind of boil down to similar principles. So I'll walk you through sort of the most straightforward way to do this. When we run our Discovery Genome-Wide Association study, we basically just run millions of regressions. And so for each genetic variant in the population, we're looking at whether it's more common among our cases or among our controls, and we can calculate an odds ratio after adjusting for things like principal components of ancestry or site where the individuals were collected or things like that. And so for each variant, we know what is the risk allele and how much does that risk allele contribute to the phenotype. Then in an independent sample, for each person in that sample, we can use the information from the GWAS, from the discovery GWAS, to basically train a genetic model, right? So if we have everybody in this independent sample genotyped, we can say, okay, at risk locus 1, this person had 0 risk alleles, so they'll get a 0 there. And at risk locus 2, they had 1 risk allele, so we'll weight that by the effect size from the discovery GWAS. At risk locus 3, they had 2 risk alleles. We'll weight that by the effect size from the discovery GWAS, and so on. And we will sum these all up, and that is that person's genetic risk score. So we can now do this for everybody in this independent sample, and then look at how that genetic risk score segregates cases from controls, for example. And we can look at whether it segregates cases from controls on the very same phenotype that we collected in our discovery GWAS, or on a different phenotype that we think might be genetically correlated with that discovery GWAS phenotype. But there are a lot of decisions that we have to make about which genetic variants are going to go into our model. So this is kind of like a feature selection step. And in general, we're not yet at a place where we have enough sample size to have a well-powered discovery GWAS and then a training set and then a testing set. So a lot of times what we do is actually just look at the performance of the scores across different thresholds within our training sets. So I wanted to show some examples of how that can be very dramatically different, and it really varies with the genetic architecture. So that means it varies depending on how many SNPs are actually causing the disease, what's the effect size of those SNPs, and how well-powered your GWAS was to detect it. So here's an example where essentially on the y-axis we're looking at the proportion of variance explained. This is actually the p-value, but it translates, it's highly correlated with the proportion of variance explained. And on the x-axis, we're looking at the threshold that we applied to the GWAS to determine which SNPs were going to go into our model. Okay, so over there at the far right, we have essentially just the genome-wide significant SNPs contributing. And as we march forward, or as we march to the left, we have gone deeper and deeper into the GWAS results, so that actually at the far left, we have basically the top half of all of our genome-wide association results, okay? And you can see that as we continue to add variation, we continue to explain more variation in our target sample. This is another phenotype with a different genetic architecture and based on a different GWAS. And here, we definitely get our best prediction pretty close to genome-wide significance. And as we go further and further into that polygenic space, now we're just adding noise. We're really not adding any more signal. And so there's always this trade-off between power and noise. And this is an example for LDL, which has a simpler genetic architecture and coming from a GWAS of over 300,000 people. This is an example of schizophrenia. And this is actually, I think now, maybe about a year old. So with the newer schizophrenia data, I think we do start to see kind of peak performance somewhere around a threshold of 0.01. So, but my point here is that there's imperfection in how well we can estimate a person's genetic liability to disease. Although that's I think a pragmatic barrier that will eventually come down as we have larger and larger sample sizes. But also the predictive power of the genetic risk score to predict disease. So that's what we were just looking at was our ability to estimate someone's genetic risk. Now we're asking, how well does the genetic risk, when estimated, predict disease? That also has some bounds on it, right? So it's dependent on the heritability and the prevalence. And this, I think, should make some intuitive sense, right? So if the prevalence is the same for two different diseases, you're better off with a genetic predictor for the one that is more heritable, right? The one that actually more of the variance is explained by genetics. So genetic liability is a better predictor of disease for diseases with higher heritability. That makes sense. And for diseases with the same level of heritability, genetic liability is a better predictor of disease when the disease is rare. And so these are just simulated data showing these principles, right? And so what I wanted to point out in this D panel here is that this corner of the ROC curve where we have, for heritability of 0.8 and disease prevalence of 1%, and we're getting very close to almost perfect prediction, that's where we actually start to approximate a Mendelian disorder, basically, where a genetic test is diagnostic. But for most of our interests and our conditions that we're studying, we're really much more in the space of heritability at the SNP-based level, so what we can explain with the common variants, being closer to 0.2 or 0.3. So this is closer to the kinds of curves we should expect for genetic risk scores alone. So I think one of the things that the field has come to realize is that a predictor doesn't have to be perfect to be useful. And we can accept that there is some imprecision in our genetic risk scores. We can accept that there is some imprecision in the predictive ability of those risk scores. And I think what we really want to start to understand is, given all of that, how can we still use this information to improve diagnosis, prognosis, treatment in an also imprecise healthcare delivery system, right? Where the time with each patient is limited, family histories are often unknown, and as was mentioned, I think a few times today, complex comorbidities tend to be the rule rather than the exception. So one of the things that we've really been pushing for is more sort of pre-translational research. And we've been trying to do this in the context of the electronic health record, because we think that this actually provides a really nice sort of translational sandbox where we can get a sense of how these predictors will perform in a real world setting, so where people are not highly ascertained for research studies, but are just coming in the door of a clinical setting. And we're doing that without actually impacting their care, right? There are actually some areas where genetic risk scores are being rolled out already. For example, I think I'm hearing from my colleagues in breast cancer that they're already being included on clinical tests, and that the genetic counselors really have no idea how to give this feedback to the patients. So we'd like to try to model these in as close to a real world setting as possible to understand what the real challenges are going to be. So we've put together this consortium, again, on the theme of team science. This leverages the eMERGE consortium, which is a network of biobanks and electronic health record data sets across about 11 different academic medical institutions. And our work group is particularly focused on psychiatric disorders. So one of the ways that we've started to look at how a genetic risk score could be useful in a clinical context was really first to understand whether these risk scores, A, would actually even correlate with phenotype in a clinical context, because the clinical phenotyping is going to be messier, and B, whether they would be specific. So we can do something along the lines of, it's sort of like a genome-wide association study, but kind of turned on its head. So this is a phenome-wide association study, where in each individual in the biobank, we calculate a genetic risk score, and then we look at how that risk score is associated with all of the phenotypes in the biobank. So we did this for schizophrenia, and these are the results. So again, a Manhattan plot, and along the x-axis we have the phenotypes grouped by category. This is an analysis of about 95,000 people across 4 different biobanks. And it's essentially a case cohort study for about 1,800 different phenotypes. And so what you'll see is that our top association here was actually anxiety and anxiety disorder. And then right below that, we have bipolar and schizophrenia. But again, we're plotting the p-values here. And so actually we're best powered to identify the association with anxiety, but our largest effect size was indeed in schizophrenia and next in bipolar disorder. But we do definitely see a pretty strong cross-disorder signal for the schizophrenia polygenic risk score. And so this is now looking at deciles of polygenic risk. And so essentially on that bottom panel, what you see is that as the decile of polygenic risk increases, we have a larger proportion of cases in our decile. And the top panel is showing the same trend, but broken down by site, so you can see that there is also some intersite variability. So that's one kind of body of evidence that we are continuing to pursue to try to determine the utility of risk scores in patient stratification. But we also are interested in seeing whether risk scores can be used to identify additional risk and resilience biomarkers for mental health traits. So a similar kind of study design, and this is very exploratory, so I will give that caveat to begin with, where we can, again, calculate genetic risk scores for essentially every psychiatric disorder in everybody in the biobank. And then we can actually look at how that genetic risk also is associated with quantitative measures of already routinely used clinical biomarkers. And I think one of the things that is particularly useful about this study design in the EHR is that we often, when we're studying the genetics of a neuropsychiatric disorder, we're doing that kind of in isolation. So particularly to get up to sample sizes of 800,000 people, which was what we had in the most recent major depression study, we really don't have a lot of additional phenotype information on them. So most of the data that, most of the additional data that's collected is usually collected on pretty small samples. And so here we're able to look across large sample sizes and test hypotheses that have never been tested before. So we did this in a proof of principle for low density lipids. And we again find that our polygenic risk score for LDL very nicely predicts LDL levels in the EHR. And then another proof of principle, we applied a polygenic risk score for coronary artery disease, which also predicted known biomarkers of coronary artery disease. So these are, again, some proof of principle studies, and we've now moved into working with major depression. We have some interesting results implicating inflammatory markers, but they're kind of not really ready for prime time yet, so I didn't want to bring them out just yet. But the goal basically is to use the polygenic risk scores in conjunction with these other clinical measures to develop multimodal markers. And I just want to finish by also mentioning that I think we have a really unique opportunity for genetics in that we can test somebody's genetics at birth, right? Not saying that we should, but we can. And the genetic risk for a disease does not change throughout your life. It is what it is from the moment that you're born. And so there's the potential for early integration into the clinical workflow to maybe shorten the diagnostic odyssey, not necessarily to supplant other ways of diagnosing, but to help kind of, I did it again, I gesticulate a lot, but to help kind of shorten the path to a biomarker that may be more diagnostic, right? So, for example, these are figures from the Rheumatoid Arthritis in America study showing that over half of people experience symptoms before the age of 35. When they asked, well, what were the symptoms that you experienced? It was things like, stiffness and wrist pain and knee pain. So the kinds of things that you would go to a doctor and they would say, well, do some stretches or take some anti-inflammatories. Eventually, these people got a rheumatoid factor test and became so symptomatic that the arthritis was visible, right? And eventually were diagnosed, a good portion of them over the age of 45, right? But if we had had a genetic risk score for rheumatoid arthritis on all of these individuals, when they first went to the doctor and said, hey, I'm having some wrist pain and some knee pain, and the doctor said, yeah, you're kind of high on the genetic risk score scale, maybe let's go ahead and order that test. This could have significantly reduced the time to that final diagnosis. And I think that's one of the unique benefits of using the genetics. Okay, so very quickly, the limitations are that individuals can have their own kind of combination of genetic risk factors. And right now, most of our genetic scores are really based on European populations, and that is a huge problem. So we really need to move into non-European populations as well. So our challenges and opportunities, I think the accuracy of the PRS to predict genetic risk is influenced by the statistical power of the GWAS, so we need to, again, continue to increase sample size. The accuracy of the PRS to predict disease status is also going to be influenced by the heritability and the prevalence. The PRS may have predictive ability when combined with other markers, but are not likely to be diagnostic on their own. We believe that they can be used, though, to identify additional biomarkers, and when integrated early into the clinical workflow, they may help to shorten the time to diagnosis and aid in differential diagnosis as well. All right, and this is my team, and I'd like to thank you again for your attention.
If there are any clarifying questions from the floor, and if not, we'll go on. Bernie and Raquel, Laurel, and all. Up to the table.
Can I just ask one clarifying question? Did you say that in the current PGC for schizophrenia that the polygenic risk threshold that was starting to come down again in terms of information, was it 0.01 or 0.1? At which threshold is it? You said 0.01,
Sorry, I think
but
I said
thought
0.01, but I think I meant
you meant
0.1.
That's what I thought.
Yeah.
Thank you.
Thank you.
Sure. This has been really interesting and a little bit more diverse than some of our other sessions. So I was just wondering if the panel had any comments about integrating some of these analyses, combining the cognitive testing with the genetics. I know, Raquel, you said you had done some genetics along with the CMB that you've been working on, and just wondering if we can talk a little bit about some of the things that you're working on independently and maybe integrating them and seeing the pros and the cons of bringing them together for stratification, enrollment, enrichment, those kinds of things.
The work in the field currently underway that is performed by consortia with data sharing is enabling this type of approach. So, for example, we are beginning a study supported by NIMH that is international, that includes 22q11.2 deletion that is associated with the 25-fold increase in schizophrenia when they become adolescent. So you can begin and follow the development from the time they're born. And the same measures are applied, and you can see that on the cognitive battery they are impaired. But the unique opportunity going from what Leah said, from the very genetic and capturing with the same methods, characterizing, for example, schizophrenia or early psychosis, and seeing, and the pattern is very similar to idiopathic. So we get from common and rare variants paradigms combined, working with geneticists and clinicians and phenotypers, applying exactly the same measures prospectively. So you can get a lot of power doing that, but it requires a decision, and there's always a give and take. You can't measure everything and agree across the sites and the investigators, what can we do well as a first step? We will follow the participants and maybe we will add measures. But some of the measures that were discussed during the past 2 days here are really amenable to cross-sites studies. And when a population like that is well characterized, data is shared, and you have multiple pronged approach, then when novel therapeutics are considered, you have a network that is ready, to talk and say, this is what we have, and is unrestricted. Everybody is in it, like the sandbox, everybody is playing together. And so I think that the field is really ripe for it now.
So let me follow up for Stephane, for everybody. So this is sort of let us imagine that we had access to all the phenotypic data that has been gathered in schizophrenia and or depression trials in industry. Okay, we had all the phenotypic data available, and there's a lot of blood stored away with potential genetic material, which I know when I was at Merck, we would do this, but we never did anything with the samples, and I don't know, they may have been thrown away by now, but just imagine it. And if you then took all that genetic material and did a polygenic risk score on that, you could look at the relationship between much deeper phenotyping and your polygenic risk scores. This is sort of an intellectual exercise, but would something like that even be imaginable or feasible or something to do? Or is that just a crazy idea?
Yeah, so I think the answer is absolutely yes, and I think one of the things that I would really like to impress on the audience here is that, I think people have been really shying away from genetics because everybody is under the assumption that you have to have massive sample sizes to do anything. That's not really true. What's true is that you have to have massive sample sizes to make the initial discovery of a SNP association with a phenotype. But there are huge consortia that are focused entirely on that. And all of that data is made publicly available. And we're really excited about people then applying those models to, more modest sample sizes that are very deeply phenotyped, exactly in the kind of design that you just described. So I think that is absolutely something that everybody should be doing.
Yeah, maybe from an industry perspective, I think we've all been doing what you've been doing at Merck, storing samples away, and projects move on and sort of never look back. So figuring out a way to do that is certainly something that I think many companies would be willing to support.
I think if I can, the only other thing I'd like to add to that is that it's never a bad investment to get genotype data on a sample. Even when the models change, even when we decide, okay, we've learned a better statistical approach for calculating polygenic risk scores, the genetics of the individual is not going to change, right? That data is there, and then go back to it over and over again with the improved models.
We're switching away from that one. Another theme that has come up, and Raquel just touched on it, is the extent to which we believe we're at a point where and Stephane, perhaps you could comment particularly as well, we're at a point or not that people would embrace some more common measures. Now, across all of our studies, whether they're NIH-funded studies or industry-funded studies. Without going into it, Stephane emphasized the variance across companies in the way in which data is gathered. Very anecdotally, I just remember visiting a site years ago when I was at Lilly, and they had 4 different sets of instructions up across 4 studies on how to administer the Hamilton Depression Rating Scale, okay? I mean, and I don't think this has been fixed, unless if I'm not sure it's been fixed. So do we have an opportunity? And then since, Stephane, you made a glancing comment on the matrix, and we sort of have it out there, Tiffany and all, but shouldn't we be open to rethinking whether that's what we want done in all studies? And if I remember correctly, there is no registration trial, I think you told me, that actually has the matrix in it, even though we invested all this time and money. So I'm highlighting this. I don't know. I mean, none of the launched compounds actually have repository matrix data we can mine. Is that correct? Because I asked that question before, and it wasn't there to be mined.
No, so it's not that people aren't using the matrix. People are using the matrix. The problem is that we don't have anything that has managed to move all the domains on the matrix to be able to get a broad claim for improvement of cognitive impairment. And in order to get that broad claim, you would have to move everything. So we've been telling sponsors that we're more open to looking at, like, okay, well, you've got to look at all of cognition still. You still have to look at all the elements. But if you think that you can move working memory, then aim for moving working memory. We still want to see that the other things don't get worse. That's important.
Okay. So the reason I'm asking this is, because of history, do we stick with the matrix? Or are we open to saying maybe we should move away I'm not advocating a particular scale, some open scale. Shouldn't that be an open discussion at this point?
Well, Bernie, and you could maybe speak to this a little bit, but I don't want to steal your thunder, but we have talked a little bit about, whether we should start organizing like a Matrix 2 working group and,
Right, yeah, I think the time is right to come back together. The Matrix initial publication came out, I think, 2005. So it's probably time to reevaluate our thinking on the matrix now that we actually have data from companies that have looked at it, and we're seeing that this is kind of a Titanic that's not really moving in total. So just different thoughts about where to go from here. So just stay tuned.
Yeah, I think it was optimistic and ambitious, but in reality, when you stop and think about it, and you think about the different domains involved, and whether or not the circuitry overlaps or anything like that, it's hard to imagine that you could have one drug that would move all of cognition. So I think that a little flexibility is warranted in that regard.
Yeah, I think we certainly would support a matrix revisited, absolutely. And there's a lot of companies.
I figured that might be the response. So.
Again, the reason I bring this up is because if we are thinking in terms of collaborative, pre-competitive things and agreeing on a common sets of phenotypic measures, we have the legacy to deal with, and transitioning from legacy is very difficult. It takes a huge alignment. On the other hand, maybe we should be open to that going forward. And again, I don't know, and that's why I was really asking but apparently nobody's done it, to take the legacy data that's on there and combine it with the genetic data. But that would be a huge undertaking. I don't know if that would be worth it, but again, it could be done, Lee. So I'm just trying to articulate a number of opportunities out there, because some of the biomarker comments said legacy data could be very useful. So I think one of the things we have to struggle with is to what extent do we argue for more interrogation of legacy data, combining some measures and seeing what we could do with combinations of measures or not. And operationally, it's a huge effort and all, versus just agreeing, going forward prospectively, we try to have some more common phenotyping tools.
I mean, I think it would be a shame to just chuck the data that we have so far. I mean, I'm sure that we can learn a lot from that as well.
Well, so I agree. So
We may
would in
yeah.
theory, we can imagine, I'm just, again, this is a mind game here, but could we imagine that there would be a way for sharing even the placebo data, where we have all that matrix data and finding about how many people actually have, how many thousands of people or even hundreds have polygenic risk scores that could be calculated, and then we could take the upper 20 tiles and see if there was a relationship with particular domains of cognition that you can pick up on the matrix, sort of testing, Raquel, some of what you have argued might be there.
But, I mean, that's sort of the point of this consortium, though, right, is to try to encourage that collaboration in a pre-competitive space and data sharing, things like that. On our end, we love to dig into the data that we have and try to find, new signals and whatever, but we don't actually see the data unless a company is coming in with an application. So that means they must have won on something before we get a hold of the data. So we don't have matrix data to look at.
But that's what I'm saying. So
Yeah.
you don't actually.
But I would
what I'm trying to say
encourage
is,
all of
so,
you to play nicely together.
So that's what I'm going to clarify. So in fact, none of the compounds that were actually went to market since the matrix had come on actually included a lot of matrix data in their phase 3 studies.
I don't believe so.
That's what you're
I
saying,
don't
which
believe
is amazing. And I like to point that out to all of our NIMH colleagues that it never made it. You would have thought, I think the belief system was that companies would embed it just in the hopes they might show something. But it's a little expensive and hard to apply, Stephane. I think what it added a lot to the cost of studies. That's what I understand.
Well, and to the burden on patients and things,
Right.
yeah.
So it didn't get done.
Plus, then there would be most of the drug registration trials that have come in since the Matrix have been for antipsychotic effect. And so if you have somebody who's acutely psychotic and then they get better, it's hard to say there's a cognition.
You mean pseudo-specificity.
Right. I don't want to say that word.
That's the history.
Earlier today, Alan provided this framework of inference to generalizability to prediction. And he said, like, the things that we do in our kind of academic labs provides inference at the group level that there's a signal there. But then when you want to move it out into a trial for Stephane, you need to be able to generalize it so that it can be battle tested and hardened like the Penn battery has been. But when you want to move to prediction, then you need your very tried and true measures. And so kind of at the risk of becoming a broken record, I do want to emphasize the importance of psychometrics. And if you look at some of psychiatry's big failures, one of them was encenicline. And there was a beautiful presentation at the Schizophrenia International Research Society not too long ago about the matrix battery and how once you start to generalize outside of the original psychometric validation studies, that things go wrong. And so Michael Green and others wrote a beautiful paper about the psychometrics of the matrix, but then when it got applied in the clinical trial study, it was tested 20 times at multiple time points, and there was no information about practice effects when applied in that manner. And so when you try to generalize from the original psychometrics and you take something that seems like a really good idea from a consensus panel, it doesn't always work well. So I would also like to volunteer for Matrix 2 and
I will say, just as a little, as a bit of a plug, it's been a real pleasure working with Raquel Gurr and that test battery and with Ruben. And the data that you presented today is going to be a treasure trove for the field for decades. And far more people have been tested on the pen battery than any other kind of cognitive test assessment and across disorders, not just schizophrenia or psychosis risk. So again, thank you for doing that work. Thank you to the people that supported it. And to Stephane, I'll just say, I'm somebody who's novelty averse. I don't like the new tasks. I'm not impressed with them until they've been battle tested and there's test, retest, reliability characteristics. And so, like, if you see a cool poster of an interesting new test, I'm not in a hurry to try to put it into a clinical trial study because, like, you know, we don't really exactly know how translatable it is. We don't really exactly know anything about the psychometrics when it's put into Slovakia. And, you know, I like having some things that are really tried and true as your go-to measures, like the pen battery, like my version of mismatch, not the 0.5 reliability version of mismatch, and many other things. So it's really important that we're here and having these kinds of conversations and learning from what works and what doesn't work, and there's a lot that we can learn about what didn't work.
So on this cognitive thing, Laurel didn't address it because we didn't ask her to, but since you brought up cognition and we like our markers to relate to clinical measures, what have we learned? What is your sense of what we've learned from ADNI? Because that was not well structured from the beginning, how we dealt with the cognition measures. Have we learned anything?
I think ADNI is interesting because it was supposed to be a fairly narrowly defined group. We certainly can tell the difference between clinically normal and clinically demented people. As even in this very carefully defined thing, there's noise. I mean, I think one of the key things that troubles me about working with cognitive testing is the signal to noise is high even in very well-controlled situations, and the heterogeneity is high. And it's hard to deal with that cross-sectionally, but longitudinally, change does appear to be more meaningful. So I'm a little dubious about the cross-sectional cognitive testing unless it's been really, really carefully validated. We have things like education, things that translate across education and cultural levels are difficult to find. And even longitudinally, the practice effect thing, I think of the first year in the religious orders study at Rush. We did some pretty careful psychometric looking at things and discovered that there was this odd pattern of people getting a lot better at one of the convents. And it turned out that Sister Scholastica was tutoring people. And I mean, who knew? We had to go to the convent and explain, no, this is not, you know, the Mother Teresa High School and you're in competition. And so you have no idea sometimes what can be happening in cognitive testing. And I'm sure everyone in this room has taken lots of tests and had tests where you did amazingly well in pure luck. I've certainly had those, and ones where you walked in the door and you did poorly. And so I think that the signal to noise in all of this is hard to deal with without really careful psychometrics. And so I guess on the other hand, the clinical variation is also subjective in ways. So again, change over time, though, in the Alzheimer's world, by the time you get to the point where you notice severe change, we can tell. I don't know. Does that help? Does that?
Yeah, I think this sets some themes, things we should follow up on. John, I'm not going to let you ask a question because I realize we should be protecting time for the last session. So unless, is that, Linda, are you in? Oh, well, I don't know if they got to say what they wanted to say.
Actually, there was one other thing that I did want to
Good
say,
point.
because I think a recurrent theme has been heterogeneity. That's come up over and over in different, in the context of evaluating different markers. And one of the things that we are also thinking about and have some preliminary evidence for is that not only can we potentially use the genetic risk score to to aid in patient stratification and things like that, but that we can also, because we can quantify this, contribution of genetic risk, you can also remove it. You can account for that variance in the measurement of other biomarkers and potentially get cleaner versions of the marker that you're trying to measure. And so that's actually something that we're kind of piloting in the lab data in our EHR right now. But I wanted to throw that idea out there because I think that it might have some legs.
Yeah, to Linda's point, please, Laurel and Stephane.
I'd like to
Raquel
follow on
comment.
that because I think we discount often the value of heterogeneity. And a wise statistician who was my mentor when I was very, very, very young, a very long time ago, Lincoln Moses, said that people think statisticians are interested in averages, but the variation is much more interesting, and that's where you learn something. So I think the fact that we're all dealing with very heterogeneous conditions is something we should take as an opportunity to learn to account for it, because that's where we'll really learn what we can do about it. So,
Stephane? And then Raquel. Any summarizing comments?
Not in particular. I think a lot have been said that covers most of what I would have said too. So that's fine.
Say that we are ready for prime time.
Okay.
I mean, there's so much that is going on in the field that we need to move, and if we move thoughtfully, we will learn from it, and, we'll open up new opportunities.
Well, thank you all. And this is going to be very, very helpful, I think, to the Neuroscience Steering Committee and in future meetings. Maybe, Linda, you can sort of take us through some next steps. Linda Harmon, maybe take us through some next steps discussions. Thank you, everybody.
thank you.